Good morning, my dear friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic order. You can also make use of it. My dear friends, in the last class, we have begun with a new topic, that is differential sexual differentiation or differences in sexual differentiation, which is also called as DSD, which was previously called as disorders of sexual differentiation. Now it is no more called as disorders, now it is called as differences in sexual differentiation. Anyway, we have seen the basics, and today exactly we will see what actually happens. So to understand that point, we need to go for embryology. We have to look into embryology. One by one, step by step, we will understand. Then there will not be any confusion. Now, we know that urogenital ridge is formed during the fourth, fifth, or sixth week of gestation. Okay, this is the first step towards the formation of the sexual organs. Urogenital ridge is formed, that is during 4 to 6 weeks. Now there are two important genes are there which is actually responsible for formation of the gonads, the sexual organs. Okay, because sexual organs may be internal, may be external, so they are formed from the gonads. Okay, so I will explain how. So that we should have the formation of the gonads first. Now two important genes are there which is responsible for formation of the gonads, maybe male or maybe female. NR5A1 and as well as WT1. NR5A1 and WT1, these are the two important genes which is basically responsible for the development of the gonads. Okay, so out of these two genes, NR5A1, so this gene is actually responsible for the development of the adrenals, and WT1, this gene is responsible for the formation of the kidney, because kidney as well as adrenals both are very important for us in the formation of the genital organs. Now till 6 weeks, 6 means almost all 1.5 months. Okay, the gonads are undifferentiated. We also say the embryogenesis is the period where the zygote is undifferentiated. It is not differentiated. Similarly, up to 6 weeks of the life, the gonads are totally undifferentiated. Okay, so which type of gonads will going to form, whether it will going to form a male one, female one, so this part is not clear up to 6th week. Okay, now that means not clear means it is not differentiated. Sex is already determined by means of the XX or XY. Already the zygote is formed, either it will be XX or XY. But development, when you see up to 6 weeks, we are not able to differentiate between the male gonads and female gonads, or no any gonads. Okay, so now suppose when there is some problem of the adrenal gland, that means the genes responsible for the adrenal gland development is NR5A1. So when there is a problem in this particular adrenal genes, then we can think of NR5A1. That means NR5, that means the genes which are responsible for the formation of the adrenals is NR5A1, and if there is no formation of the adrenals by the age of 6 weeks also, by the time of 6 weeks also, then we can anticipate there may be some problem related to adrenals. Similarly, if there is no formation of the proper kidney in undifferentiated form, then again we can think of the genes like WT1 is get affected. Okay, now so these two genes are basically responsible for formation of the gonads. Okay, so once the gonads are formed, okay, now the next step is whether gonad has to be become a male gonad or whether gonad has to be become a female gonad. So because differentiation is still not over, it's yet to take place. So now gonads are formed. Now the next is either it has to become a male gonad or either it has to become a female gonad. This is the next step. Now Now suppose this differentiation, male or female, this differentiation is basically done by a gene by name SRY. SRY. So what is the name of this gene? Sex determining region of the Y chromosome. Okay, so this is very, very important. Now the gonads, whether they become male or may become female, so who will going to determine it? The person, that means the point which will going to determine this particular point is SRY gene. The full name of the SRY gene is sex determining region of the Y chromosome. As the name itself suggests, this is only present in the Y chromosome. So this is only present in the Y chromosome. So which is supposed to be there in the Y chromosome. If XY is there, in that particular zygote, this particular gene will be there, and next differentiation will going to start. Okay, then what about the female? No, the female we will explain later what actually happens, because the main point is this one. Okay, now suppose if this, let us take let us take a zygote is there which contains SRY, probably this is a male one, so it contains SRY gene. Then what happens? If this gene is present, then this gene will going to send the downstream signals. Okay, so this gene will going to produce some of the downstream signals, like for the production of NR5A1, SOX9, etc. Okay, so because of signals from these particular genes, okay, there is production of some of the substances like NR5A1 and SOX9, etc. Now what is the function of these substances? These substances will go into form the proper testes from the undifferentiated gonads. They will go into form the testes from the undifferentiated gonads. Till 6 weeks we know that it is undifferentiated gonads, and after 6 weeks all these happenings are taking place. And now what happens? These two substances are formed, and because of that the undifferentiated gonads are now get converted into testes, because this is Y gene, isn't it? Therefore it is converted into testes. Now once the testis is formed, now this testis contain two types of cells. One is Sertoli cells and another one is Leydig cells. Leydig cells and Sertoli cells, that we know. Now what is the function of the Sertoli cells? The Sertoli cells, they will secrete one important hormone which is called as anti-Mullerian hormone. Okay, Sertoli cells, they will go on to produce the anti-Mullerian hormone, and this will lead to inhibition of differentiation of Mullerian structure. Sertoli cells will secrete the anti-Mullerian hormones, and this will lead to inhibition of differentiation of the Mullerian structure. Now in the fetus, when it is developing, embryologically when it is developing, okay, there is all the possibilities are open. It may be get converted into the sexually, it may be get converted into the Mullerian duct or Wolffian duct. But if it is Sertoli cells are there by the testes, in Sertoli cells they produce anti-Mullerian hormone, then what happens is the possibility of formation of the Mullerian structure will be inhibited. There is no further possibility of formation of the Mullerian structure, because one of the hormones which is produced by the Sertoli cells is anti-Mullerian hormone. So now there is no formation of the Mullerian structure. So if there is no formation of the Mullerian structure, then there is no formation of the female internal sexual hormone, the sexual structures, genital structures. Okay, so Mullerian structure usually give rise to the development of the uterus and fallopian tubes. Now as these cells which are hormones which are produced from the Sertoli cells of the testes will produce a hormone called as anti-Mullerian hormone, and this hormone basically blocks the differentiation of the Mullerian structure. So possibility of formation of the Mullerian structure and thereby uterus and fallopian tube is totally blocked. Now what happens? Second cell is called as Leydig cells. The second type of the cell is Leydig cells. So this again produce two important substances. Okay, So we are talking about SRY, Y chromosome. In case of males, we are talking, and in the males, all these happenings are taking place. Now, the second type of the cell which is present is Leydig cells. Leydig cells, they will secrete two important substances. Number one is testosterone, and another one is insulin-like substance 3. Okay, insulin-like substance 3 and Leydig and testosterone. These are the two important hormones which is produced by the Leydig cells. We know that what happens by the Sertoli cells. Sertoli cells, they produce the antimullerian hormone. But Leydig cells, they produce the insulin-like substance 3 and as well as testosterone. Now, this secretion is due to the action of SCG on LH receptors present in the Leydig cells. How these Leydig cells produce these two substances? Leydig cells produce these two substances or these two hormones, you can say, due to the action of SCG, human choriogenic gonadotrophin hormone, SCG on LH receptors, which are present on the cell surface of the or cell membrane of the Leydig cells. Now, okay, testosterone is produced, insulin-like substance 3 is produced. Now, what happens to testosterone? What the testosterone will do? This will be the game changer. Okay, this is the actual game changer. What happens? Now, the testosterone is get converted into another next form, which is called as dihydrotestosterone. Okay, testosterone immediately get converted into its next form, which is called as dihydrotestosterone, and this conversion is possible only in the presence of an enzyme which is called as 5-alpha reductase. Very important enzyme. Okay, 5-alpha reductase. Okay, now it means the testosterone is now converted into dihydrotestosterone by the help of a 5-alpha reductase enzyme. What happens to insulin-like substance? What is the function? Now, the insulin-like substance will now going to produce the testicular, which is helpful in testicular descent. What do you mean by testicular descent? In the beginning, where the testis is formed in the fetal stage, it is an abdominal organ. Okay, now it has to come down to the scrotum, isn't it? Now, there will be a descent, gradual descent of the testis from the abdomen to the scrotum. Okay, so now the enzyme, rather hormone, which is responsible for the descent of the testes from the abdomen to the inguinal ring, okay, to the inguinal ring, is by means of insulin-like substance 3. What I mean to say is, actually, testosterone and as well as insulin 3 like factors, both are helpful in the testicular descent. But from the abdomen to the inguinal ring, it is majorly the function of insulin like substance 3. Okay, so insulin like factor 3 helps in the descent of the testis from the abdomen to the inguinal region, while descent from the inguinal region to the scrotum is majorly the function of adrenal hormones. Okay, so adrenal hormones are responsible for descent of the scrotum from the inguinal ring to the scrotum, whereas till inguinal ring from the abdomen, it is a function of insulin like substance. Now, so this gives a clinical clue here. If we find that abdominal testis, suppose the testis is still in the abdomen, so it suggests that testosterone is not formed properly, or testis itself is not formed properly, so which basically indicate a testicular dysgenesis. If you find the testis in the inguinal canal, it just suggests that the testosterone is normal and the problem is with the adrenal glands. That means if the testis is in the inguinal canal, that means Leydig cells are producing the normal functioning. Leydig cells are normally functioning and testosterone and insulin like factors are produced. But still, if the testis is not there in the inguinal canal, so that suggests that Leydig cells are creating some problem. But if it is there up to the inguinal canal but not in the scrotum, then it is clearly the problem related to adrenal gland. Now the testosterone will form the Wolffian duct. So we know that Sertoli cells and Leydig cells, they produce the testosterone. Now the testosterone will form the Wolffian duct, and Dihydrotestosterone helps in the formation of the penis and the scrotum. Now the further development takes place. Testosterone is already produced from the Leydig cells. So now testosterone, which is already produced, that will form a duct called as Wolffian ducts. Now there is no chance of formation of the Mullerian duct because Mullerian duct has been already blocked by the Sertoli cells, isn't it? Anti-Mullerian hormone, which is produced from the Sertoli cells, they already block the formation of a Mullerian duct. So there is no chance of formation of the Mullerian duct here. Okay, therefore there is formation of the Wolffian duct. And the next form of the testosterone, that is dihydrotestosterone, that helps in the formation of the penis and as well as scrotum. Okay, penis and as well as scrotum. So for Wolffian duct formation, we need testosterone. Very clear. 2 points are there. If the testosterone is there, there will be formation of the Wolffian duct, and if the testosterone is there, there is no formation of the Mullerian ducts. That means if the testis is formed, rather than testosterone, if the testis is formed, there will be formation of the Wolffian duct, and if the testis is formed, there is no formation of the Mullerian duct. The reason is very simple. One of the cells of the testis, that is Sertoli cells, so they will going to produce the anti-Mullerian hormone, and which will going to block the formation of the Mullerian duct. Now, but for the formation of the both internal and external genitals, now Wolffian duct is formed from the testosterone. Now for the formation of the internal and external genital organs, okay, the concentration of the hormone is very important. See, everything may be fine up to this point. Now the next point is we have to form, there should be formation of the external and internal genital organs. Now what determines the proper formation of this process? That is the hormonal concentration. Which hormonal concentration? Hormonal concentration of the specific hormones. Okay, specific hormones. Specific hormones, as we are discussing about the male, specific hormones is testosterone and dihydrotestosterone. For example, for the formation of the epididymis, that is internal genital organs in the male, epididymis, vasa deferens, seminal vesicles, the local concentration of the testosterone hormone should be very, very high. If you want to have a proper formation of these internal genital organs of the male, testosterone concentration should be very, very high. That means it should be in a proper level. And for the formation of the external genital organs like scrotal bag, then urethra, then penis, okay, etc., we need to have dihydrotestosterone in the normal level. So both the forms are very important. If the testosterone is normal, then what happens? The internal genital organs of the male will be properly formed. If dihydrotestosterone is normal, then the external genital organs of the male will be properly formed. Okay, up to that point everything was normal, but unfortunately there was no concentration of these two hormones. Then in spite of having everything normal till that point, formation of the testis and all, and Wolffian duct and all, but if these two concentration of the hormones are not proper, again there will not be proper formation of the external and internal genital organs in male. Okay, now to act on the adrenal receptors along with the testosterone, both the testosterone and dihydrotestosterone will act on the adrenal receptors. Now this is one more important thing. Now we have got two things, isn't it? Testosterone and dihydrotestosterone. One of the peculiarity of this, speciality of these two hormones is, so both of them can act on adrenal glands because they have got some receptors there. Okay, so this testosterone and dihydrotestosterone, then they can go to the adrenals and they can bind the receptors present on the adrenal cells and they can show their effect there. So if dihydrotestosterone is not there, then external genital organs will not be look like a normal external genitalia. What is the role of adrenal gland here? So that I will explain. So this point is repeated. External genitalia will not looks like a normal external genitalia if there is no proper dihydrotestosterone level. Internal genital organs will not look like a normal internal genital organs if there is no proper level of the testosterone hormone. Now, okay, so I have not discussed the role of the adrenal gland here. That it is little confusing. I will take it separately. You just understand up to this level. Now, what happens in case of the females? Isn't it? What happens to the females? We have only discussed about the male. Now, how the undifferentiated gonads are converted into ovary. Gonads were converted into testes, isn't it? Why they get converted into testes? Because there was a gene which is called as SRY gene is there, and that is responding some of the downstream signals, and because of that there is formation of the testes. Now suppose in case of the female where there is SRY gene is absent, because it's not Y, it is only X, isn't it? So there is no Y, so this gene itself is absent. Then what happens? Then the undifferentiated gonad which was present there, that will not be converted into testis. Then what happens? Just like male gonad differentiation, the genes like POXL2, VNT4, DAX1 and RSPO1 etc. are required for the female sexual differentiation or female gonad formation like ovary. Okay, just like SRY is there for the formation of the testis. Similarly, for the formation of the ovary, we need some separate genes like POXL2, WNT4, DAXC, DAX1, RSPO1, etc. These are present in the X chromosome. These are present in the X chromosome. The problem comes now there is 2X is there. So whether the genes are coming from the both the X, of course they may come, but usually what happens, both the X chromosomes are not usually required. There is a Mendelian hypothesis, a hypothesis there. Usually by this time one of the X will be inactivated and only one will be active. Inactivated in the sense it is working up to 20%, 30% only, not dead completely, inactivated, it is suppressed. Only one X will be dominant and that is producing the genes. So that we'll discuss in Mendelian hypothesis. Okay, so now one X is sufficient to produce the ovary, and the ovary has got, so because of these genes which is present in the X chromosome, the ovary is produced. In the ovary there is a cells called as theca cells. The cells are called as theca cells. Another type of the cells which are present in the ovary, they are called as granulosa cell. Theca cell and granulosa cell, these are the two types of cells which are present in the ovary. So theca cells will produce androstenedione, while granulosa cells will secrete the estrogen. Okay, so now just like there the Sertoli cells and Leydig cells were there, here the theca cells and as well as granulosa cells were there. Now here the androstenedione will be produced by the theca cells, and granulosa cells will go on to produce the estrogen. However, this is not the main source of estrogen. Initially it produces estrogen. The main source of estrogen is conversion of androstenedione into estrogen by the action of aromatase in the adrenal gland. See, just like in case of males, where the testosterone is converted into dihydrotestosterone by the help of one particular important enzyme, reductase. Similarly, here also, the androstenedione which is present, so that will be converted into estrogen by means of a very important enzyme called as aromatase. There it is alpha reductase, here aromatase. Now what happens? If aromatase is absent, suppose if the aromatase is absent, then there is no production of the estrogen and androstenedione level will raise in the blood. Got it? Just like testosterone is converted to dihydrotestosterone, similarly the this will convert into androstenedione will get converted into estrogen. Suppose if this conversion is not taking place because there is absence of the aromatase enzyme, then what happens? Androstenedione level will going to raise in the blood. Therefore, this is one of the virilizing hormone. This is one of the virilizing hormone. So in female, if there is no SRI gene, hence there is no formation of the. So in female, there is no estrogen, hence there is no formation of the ovaries. So in females there is no anti-Mullerian hormone, hence there is no formation of the Mullerian duct, which further leads to the formation of the fallopian tubes, uterus and upper vagina. Okay, so there is no testosterone also, so there is no formation of the Wolffian duct formation, and there is no formation of the lower vagina and clitoris and labia major. These are the things which happen because of the virilizing hormone. Okay, so if this aromatase enzyme is absent, all these possibilities are there. Now, testosterone is converted into dihydrotestosterone by the 5-alpha reductase enzyme, as I told earlier. So we know that testosterone forms the internal genital organs, while dihydrotestosterone will go into form the external genital organs. And if there is a deficiency of the 5-alpha reductase enzyme, there will not be any conversion of the testosterone into dihydrotestosterone, and there is no formation or underdevelopment of or under virilization of external genital organs in the male. But internal genital organs may be right because testosterone is there. So these two enzymes are very important. One is 5-alpha reductase enzyme in case of males and aromatase enzyme in case of the females. So if these two enzymes are absent, see, aromatase enzyme, it is absent, so then exactly the opposite will going to take place. Okay, so there is no estrogen, so hence there is formation of the ovaries. And so these are some of the basics. In the next class onwards, we will going to see the continued portion of the same. Okay, thank you very much.